Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Advancing Genomics into Personalized Medicine. Ever since the completion of the Human Genome Project in 2003, the goal of scientists and medical researchers has been to take genomic information and use it to develop new diagnostics and therapeutics. Significant strides have been made in this effort, but a number of challenges remain, including data analysis and deconvolution. We've assembled an expert group of panel members today to address this issue during the webinar. Let's meet them. Dr. Craig Webb is a professor at the Center for Cancer Genomics and Computational Biology at the Van Andel Research Institute. Craig is going to provide a two-part detailed analysis of personalized medicine and discuss his ideas for moving this field forward more quickly and efficiently. Dr. Yuri Nikolsky, Vice President of Research at Thomson Reuters, will explore methods of analyzing omics data for applications in personalized and translational medicine. And Dr. Cornelia Polyak, who's an Associate Professor of Medical Oncology at the Dana-Farber Cancer Institute, will examine the challenges and opportunities for the personalized treatment of breast cancer. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So if everyone's ready, let's get going. Dr. Craig Webb will be our lead-off speaker. Craig, the floor is yours.
Thank you, John. Well, it's my pleasure to talk to you today about our road towards personalized medicine, and this is my contact information if you'd like to contact me. Next slide. I'll be laying out my presentation in two parts. I'll start out with a brief introduction to genomics, personalized medicine, what a biomarker is, and also tell you about the institution that I reside at, and then focus a little bit on some infrastructural elements that we've built over the last decade to allow us to actually practice personalized medicine. And then I'll break, and then at the end I'll come back to some examples of our application of this infrastructure towards real patients with late-stage cancer, and then end on the future. Next slide. So this slide really just tells you that our path towards the real-time application of personalized medicine is a long one. So we've spent 6 years developing the infrastructure, which I'll touch on, to allow us to actually treat prospective patients in a real-time fashion. But I also do maintain a lab that's focused on tumor metastasis and understanding the molecular basis of metastasis. Next slide. This is just to illustrate one application in the lab. This is actually looking at, again, using GeneGo technology, looking at the systems that we believe to be involved in the metastatic phenotype, in this case in pancreatic cancer. And so we use a systems approach to identify potential drivers of metastasis. In this case, we believe there's a histamine signal being produced by Kupffer cells in the liver that enable pancreatic cancer metastasis. But I won't be talking about this aspect of our research. Next slide. So first, an introduction to personalized medicine in general. And this is the Van Andel Institute in the middle. That's a picture of the institution, and these buildings around us are within a one-block area being built over the last decade, about $2 billion of local money being invested in life sciences and healthcare, including medical schools, cancer centers, and the like. Just to let you know that Grand Rapids is serious about life sciences in general. Next slide. Recently, we are affiliated with the Translational Genomics Institute out of Arizona in Phoenix, and Dr. Jeffrey Trent is the research director of both institutions. Next slide. Both of us have really been focused on the product of the Human Genome Project, and Dr. Trent himself worked with Francis Collins there on the right, next to Craig Vandler and Bill Clinton, on the sequencing of the Human Genome Project. Next slide. The challenge, of course, as we all know, is there's a vast amount of gap between data, the amount of data that's out there, and it's getting worse with the likes of next-generation sequencing, and the abstraction of data to information, to knowledge, and then clinically useful information and knowledge, which is minimal. So our challenge has been, over the past decade, to try and close this gap. Next slide. Case in point, if you do a literature search, is the peer-reviewed articles looking at cancer biomarkers, close to 200,000 publications. yet very few of those, of course, have actually made their way into clinical practice. Next slide. So one of the products of the Human Genome Project, of course, is that we understand genomic structure. We understand human genomes have less genes than grapes, so clearly the complexity does not occur at the number of genes. It occurs at the systems level. On the right here, of course, is a classical systems diagram, and that's obviously the focal point of Thomson Reuters GeneGo, Metacore products, and the like. And we model these systems quite actively in the lab. Next slide. Obviously, a lot of technologies have evolved through the years at the chromosomal, DNA, RNA, proteomic, and metabolomic levels. I'm not going to dwell on these. These are somewhat outdated now. And next slide. Moving towards the beasts of next-generation sequencing that are generating billions of data points and require supercomputing to analyze the data. And we'll talk a little bit more about how we're doing that. Next slide. Now, coupled with the fact that the cost of cancer care and the health economics and economics in general is a big area, topic area, is increasing, especially for end-of-life patients where majority of healthcare costs are actually spent, is actually at the end of life, the last 6 months of life. And so this is a real problem in healthcare, which we need to resolve. Next slide. So the hope is this area of personalized healthcare, the idea that we'll be using molecular information or biomarkers, which we'll get to predict disease, detect disease early, accurately diagnose and prognosticate disease, and then treat the disease with the appropriate therapy, given the molecular information of the disease in the patient. The idea being that we reduce this disease severity over time, and disease severity over time, of course, equates to healthcare costs as well. So all the stakeholders, if you like, benefit from this personalized approach. It's a bad term, but we really use terms like molecularly guided or precision medicine, but personalized medicine is a name that's stuck. Next slide. So just some definitions of biomarker. The key word here is an objectively measured thing, whether it could be blood pressure, it could be an image, it could be a chromosome aberration or overexpression of a protein or a gene expression signature or a sequence variant. But it's a characteristic that's objectively measured as a measure of disease or a pharmacological a therapeutic response to an intervention. So that's the FDA definition of a biomarker. Again, objective is the key word. Next slide. Personalized medicine, of course, is using these biomarkers to achieve this idea of personalized healthcare. Next slide. Now, the area that we focus most of our efforts on is this idea of aligning the right drug to the right person at the right time, i.e. of pharmacogenomics. So, as we all know, patients with the same, quote, anatomical disease do not respond the same way to a given therapy. So we're trying to maximize therapeutic index by aligning the patients to the right drug to avoid toxicity and maximize efficacy. Next slide. There's been many examples of this, or a few examples of this, I should say, and most of these recently, whereby targeted therapies have been aligned with what we call companion diagnostics. And when you actually identify the driver or vulnerabilities within the tumor, if you like, the driver mutations that cause the cancer or the progression of the cancer, with the right drug, we get greatly improved responses. And here's some examples with Gleevec or Herceptin or the Plexicom agents or Arisa, for example, in these different indications. Next slide. Now, the issue with that is that each of those companion diagnostics took a long time and millions of dollars to develop, nearly as long as the agents in many cases. And so the number of drugs that are out there that could be available to this approach of a line of companion diagnostics or pharmacogenomics are staggering, and there's just some knowledge bases that we use to mine the world of the drug universe and the targeted mechanism of action of these drugs, and including GeneGo at the bottom left there. That's looking within MetaDrug and looking at the targeted mechanisms of action of certain agents. And so these are some of the knowledge bases that we use for this. Next slide. So the 50,000-foot view of what we're trying to achieve is that we're deeply profiling individuals and their disease using various molecular technologies, aligning that molecular fingerprint, if you like, or profile with a historical perspective of the patient's illness, and we actually do a lot of electronic medical record integration at the Van Andel. And then through a magic black box, which I will describe in the second part of the talk, align the right therapists to the patient. It sounds easy, but obviously it requires infrastructure. So The critical infrastructure we set about building a decade ago included informatics, included a clear certified molecular diagnostic lab, and a clinical trial alliance, which I'll talk about very briefly. Next slide. So the translational research enterprise we've built is simplistically, this is a 2000 slide actually, talks about, we start actually with biospecimens, this idea of biobanking in the top right here. It's critical that you don't just get specimens, that you get the clinical data with the specimen, not just a pathology report, but information about how the patient was treated and how the patient responded, outcomes, for example, obviously. And that allows us to, over time, bank enough material to retrospectively discover novel biomarkers or approaches to predict best therapies for these patients, and ultimately apply those back to the clinic in a prospective trials, which requires things like a molecular diagnostics lab, a clear certified lab, but also clinical trial infrastructure, which we developed here, which we called Connexus. So, and in the middle there, arguably the most important component is the informatics, or we call it translational informatics, that allows us to exchange data, information, and knowledge across the clinical and research enterprises. Next slide. So just to quickly show you some screenshots. So this is our product that we developed actually as a commercial product called Xenobase Biointegration Suite that allows us to track information about patients or subjects in general, samples, experiments, and also analyze the data within the same setup. So you can query the database, its interfaces with electronic medical records, and also allows us to perform integrated analysis of phenotype and genotype, which I won't dwell on too much, but we've used this extensively. Next slide. It also allows us to create interactive and actionable reports for physicians that they can act on. And on the left here, you'll see more of these in the second part of my talk, how we use these. And then probably the most important aspect is that we also can track patient or subject outcomes, so we know whether our predictions are actually matching up with the actually observed outcomes. Next slide. Very briefly, we did form a high-complexity, clear-certified lab with a local hospital, Spectrum Health. We offered FDA-approved tests back then. It was the AmpliChip, for example, from Roche, but also the cell-circulating tumor cell system from Veridix, but also offer some homebrew diagnostic tests that we developed initially back then on the U133 2.0 Affymetrix platform. Ultimately, this was sold to Sequenome, who purchased this lab and actually run their CLIA lab in Grand Rapids, Michigan, as we speak. Next slide. We also formed very quickly here the Clinical Research Alliance, a multi-institutional alliance in West Michigan with hospitals and private practice groups, primary care doctors, etc. We became actually members of the Critical Path Institute and have actually helped qualify some novel biomarkers through the FDA qualification process, but I won't dwell on that here. Next slide. But the main point is that you really need, those are the three legs of the stool that you absolutely need to allow you to actually treat patients in real time. You need, obviously, a clinical trial infrastructure to consent patients and collect samples. You need a high-complexity CLIA lab to actually profile patients using these latest technologies, and you need the informatics to actually be able to integrate the data and actually provide actionable reports to patients. So I will hand this back. Thanks, John, and look forward to the next part of my talk.
Craig, thanks so much for that wonderful overview of personalized medicine. And as Craig noted during his presentation, he's going to be back to give part 2 right before we begin the question and answer section of our webinar. And if you're just now joining our webinar, welcome to you all. As I mentioned a second ago, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Dr. Yuri Nikolsky, will now begin his presentation on analyzing omics data for personalized and translational medicine. Yuri, you can begin your presentation.
Thank you, John. I would like to talk about the methods of functional or systems biology analysis, which is used in personalized and translational medicine. Next slide, please. On this slide, we just show that the translational research and personalized medicine to a large extent are based on analysis of omics data coming with patients, meaning next-generation sequence data, SNP data, personal genotyping data, expression data, proteomics, and so on. Next slide, please. To analyze such data, it's not sufficient to use statistical analysis or old methods of molecular biology because we have to deal with many genes, many thousands sometimes genes and features at a time. So for that, we need to apply systems biology tools, and systems biology is believed to be the main research engine for this kind of research. Next slide, please. There are several definitions of systems biology and many ways to describe it. I really like the definition done by Lee Hartwell back in 1999 in his Nature paper. He's a biologist, and he believed that molecular biology needs to be understood in modules, and the modular organization of cells follows from proteins working in groups and complexes and pathways. And this is essentially what modern methods of systems analysis, knowledge-based functional analysis, based upon. Next slide, please. Essentially, for data analysis, there are two methods which broadly used for systems of functional analysis, and one is based on enrichment by category or ontology entities, and another one is based on enrichment with protein interactions. So this is shown on the slide. Let's say we start with a list of genes which are shown as red dots on the left top corner, and those are genes which are coming from omics assays, let's say overexpressed genes or genes with important SNPs. So there are two ways to analyze this list. One, it can be ran against a list of genes with known functions, such as pathways process, like gene ontology process, for instance, drug pathways or pathways used in commercial programs for analysis. So essentially what you get out of it is a relative ranking of your genes compared to the known pathways and processes. It's quite important, but it's not very high resolution method of analysis. You cannot, for instance, guess potential drug targets just from knowing relative distribution of pathways in your list. So to do that, you need to apply a database of protein interactions, which is shown on the right side, and look for relative enrichment of every gene and protein in interaction relative to the complete interactome. And the assumption here is that the proteins with more interaction than you would normally expect, enriched interactions, are probably more important for the disease or for the other phenotype. And it can be done simply by ranking all the proteins based on the number of interactions with your list of genes of interest, or the interactions can be packed into networks, and here you need to apply algorithms and filters to be specific. Next slide, please. To understand this process in more detail, we probably need to start with a visualization of proteins and interactions on pathways and networks. Usually the visualization done in the form of nodes like proteins and edges. Edges show connectivity between the proteins. It's very important to know that proteins are connected through these binary links, but probably it's not sufficient to know, and there are several levels of complexity which need to be covered to do really meaningful and deep analysis. Next slide, please. The first level is to show directionality between the proteins, directionality of interactions. Protein A works on protein B, but not vice versa. And in human or high eukaryotes, every protein interaction actually can be presented as a directed binary link. And when we have this representation, for instance, we can use the methods of directed graph, which provide the network connectivity and network algorithms. Next slide, please. The next level is effect. So some protein interactions are positive and some are negative. So we have activators and we have inhibitors. So that has to be shown as well if such details can be picked up from the literature. Next slide. So the next level is the mechanisms for interactions. They are all different. Transcription factor binding to promoter of target genes is one type of interaction, and physical binding is quite different. Then we have metabolic and transport reactions and so on. So that has to be reflected on the interaction links. Next slide, please. And proteins have different functions. There are ligands and there are receptors, there are kinases and phosphatases, transcription factors, effector proteins. Altogether, there can be 30-35 different functions which can be distinctly shown on the pathways. So when we have that rich semantics with directionality, effects, protein functions, and protein interaction mechanisms, we can actually do quite sophisticated analysis. Next slide, please. So the interactions connected into longer chains of interactions known as pathways. In human and higher eukaryotes, signaling pathways represent linear chains of consecutive binding and reactions and some metabolic transformations. 11 steps on average, and they overlap in signal transduction parts. So the pathways very important to be used for prioritization on high-level networks. Next slide, please. It works pretty much like GPS works. There are many ways to get from point A to point B using all kinds of roads, smaller roads, larger roads. But usually people use something like shortest time use of highways. And that's exactly how the pathways are applied for building networks. Next slide, please. So this is one of the examples. So this is a network built out of overexpressed genes in some cancer data. And the thick line on this network is the pathway coming from what's known as canonical signaling pathways, most likely direction for information transfer. Next slide, please. So the pathways on its own are connected into what's known as pathway maps or wiring diagrams to depict biology in high-level terms. So you can think of KEGG maps, for instance, for such a presentation. In Metacore Thomson-Reuters system, there are more than 1,000 pathway maps, for instance, for signaling metabolism and pathology for different diseases. Also representations for high-level networks, pre-built networks, and proteins and complexes. So these structures are broadly used in enrichment analysis in ontology terms. Next slide, please. So this slide shows the analytical workflow in functional analysis or systems biology analysis. Essentially, it's all about filtering the list of genes and coming up with the genes which are most important, such as potential biomarkers and drug targets. And the methods of systems analysis applied consecutively. So we start with experiments which can be pre-filtered using different parameters such as ortholog, species, process, disease, and so on. On the next level, enrichment analysis for the list is done using different tools. For instance, gene set enrichment analysis, hypergeometric distribution, page, and some other algorithms. And multiple ontologies can be applied at this level. KEGG, for instance, gene ontology for process, or some ontology built by companies like Thomson-Reuters for canonical pathway maps, disease biomarkers, toxicity biomarkers, and so on. So this level of analysis allows to narrow down a list of interest to something which is more specific, for instance, apoptotic genes or cell cycle genes. And the next level, which we call interactome analysis, we can define the most over-connected first and second level interactions of proteins. And this way we can identify the most important transcription factors and kinases, proteases, and so on, proteins of different functions, which are linked to the list of interest by the number of interactions. And finally, we can do network analysis using multiple tools. Next slide, please. In modern systems or platforms for systems analysis, the workflow quite easy to use and automated. The user can start with any source of data of interest. It can be expression array, it can be next generation sequencing data, list of proteins from proteomic profiles, can start with chemical structure for small molecule compounds or just interaction data. And then the analysis proceeds in automated workflows or tools used one by one, tools for enrichment, interactome, and network analysis. Next slide, please. Now I would like to describe probably in more detail a couple of applications of these tools for functional analysis specific for oncology assays and with applications in translational and personalized medicine. So there are quite a number of ways that patients can be tested at gene level, epigenetic level. Expression profiles can be taken, proteomic profile, metabolites from body liquids, and so on. So what we know about all this testing and multiple data types is actually three main things. One is that no single level of data can provide with sufficiently accurate biomarkers. If you have just gene expression, we can come out with gene signature multivariate biomarker. But in many cases, gene signatures are not working quite accurately, and their predictive power actually diminishes with the number of experiments. Also, the data set assayed at different levels do not overlap quite well. For instance, if you do mutation analysis of SNPs and we have a list of overexpressed genes from the very same patient, most likely very few genes would be common on these assays, both mutated and overexpressed. So you cannot really compare them directly, and statistical methods of correlation which are currently used are not really adequate for that. So next slide, please. How can we compare this very important assay data coming from DNA, RNA, protein level and make sense out of them? There are several ways to do it using functional systems level. For instance, we can do what we call synergy in enrichment analysis and look for the pathways and cellular process which would be both hit by the data from different data sets and data types. We can use interactome tools to come up with, for instance, a list of transcription factors which will be mutated with a number of overexpressed genes in different type. We can do this analysis in one or multiple steps, and we can also build what's known as causal networks, so essentially reconstructing the disease pathology using different types of data, genetic data, expression data, proteomics, and so on. Next slide, please. On this example, we show how some of these methods work. So here is comparison of mutated and amplified genes in breast cancer, which we published a few years ago with Cornelia Polyak. So in breast cancer, in this study we identified about 1,100 of somatic mutations, and also 1,700 genes or so were amplified in one of 30 amplicons. So if you look at these two types of data, very important genetic changes happening in breast cancers, and look at the intersection of these data sets, you'll see that intersection is actually very small, statistically significantly smaller than expected on random. So how you compare that? Well, on the next slide, you can see that the pathways can be used as template for mapping both types of data. So on this pathway map for extracellular remodeling, matrix remodeling, the genes in red squares are amplified in one of many amplicons in one of many patients in breast cancer, and the genes in blue squares are mutated in one or multiple cases. So you can see that the genes are either in red squares or blue squares, but they do belong on the very same pathway. So this is a complementarity or functional synergy which can be captured by different methods and which is quantitative. So for the large data sets, we can come up with a very simple tool which would calculate this synergy, and that can be calculated between SNPs and expression data, between mutations and amplifications, or any other multiple types, which is very handy. On the next slide, I have an illustration showing what relative connectivity in interaction means. So let's say we look at a certain data set of 320 genes or overexpressed genes or proteins, and we look at specifically P21. So P21 has more than 200 interactions in entire human proteome of 24,000 proteins or so with known function. So as a proportion of that, for the data set of 300 genes or proteins, you would expect it to have 5 interactions. But sometimes it has less, or sometimes it has many more. So if it has more, it's over-connected and probably more important for the data set. And over-connectivity can be calculated in different ways, and p-value can be calculated as well. So this method, a very simple one, allows to rank transcription factors or kinases or receptors based on the number of protein interactions for the set of interest. Next slide, please. So such analysis actually can be extended not for just the first level neighborhood based on interactions, but through the entire proteome of 24,000 proteins. So each protein can be evaluated and the number of interactions or pathways, the other pathways, connecting it to the list of interest. So we start with differentially expressed genes, and build what's known as shortest path network between all of them. And this kind of network involves pretty much all the genes in human 3 or 4 steps. So for each gene, we can calculate the number of pathways coming through it, and overconnected pathways, overconnected genes would be considered as topologically significant genes. Next slide, please. Topologically significant genes are very complementary to the experimentally shown genes in such a way that they reconstruct signaling, which is quite often missed from expression profiles or proteomic profiles. So a calculation of these topologically important genes then allows to reconstruct the upstream signaling, which is very important for defining drug targets, for instance. On the next slide, we show such synergy in one of the pathway maps for regulation of lipid metabolism. So some of the genes, enzymes, are circled in red. So they came from proteomics and differentially expressed genes sets. And the genes in blue in the nucleus, transcription factors, are deduced as topologically significant regulators. So you can see that the genes coming from a profile itself and topologically significant one are pretty much complementary, and such complementarity can be calculated. Next slide, please. So the culmination of this analysis is probably construction of causal network, which are a bit more comprehensive, and they can be divided into triggers, which is ligand receptor pairs, relevant signaling pathways for signal transduction, a set of transcription factors which co-regulate effector genes, which define the phenotype. So such networks can be built for individual cancer patients, for instance, to come up with potential drug targets. Next slide, please. And these drug targets can be calculated as most important hubs, for instance, or influential genes on causal networks. This is something which is actively used in our service contracts with pharmaceutical companies and universities. So the next few slides deal with using the pathways as templates for functional biomarkers. Next slide, please. On this slide, we depicted the common idea about using omics data, in this case expression data, for coming with gene signature or multivariate biomarker, a set of genes which would be most characteristic for a condition, for instance, for predicting certain effects of drugs or viability after certain treatments in cancer. Next slide, please. So unfortunately, this simple model of identifying the best overexpressed genes deriving the condition does not work very well. So this is an illustration from Nature Biotechnology paper published by FDA researchers last year. And this is a result of about 2 years old study known as QC2. In this study, multiple data analysis teams presented on the left were given the same data sets for cancer data sets and 2 toxicogenomics data sets. And every team and teams were selected from universities, from top pharmaceutical companies and also specialized software companies. So all these teams were to come up with the best possible models for generating gene signatures. And in this table, the performance of the signatures is depicted. So what you see here in colors is Matthew correlation coefficient, which shows performance of different models on different data sets. So the bottom line here is that quite a number of teams came up with models which work well on a simple phenotype, such as gender or ER status in breast cancer. But when dealing with complex data, complex phenotypes, for instance, of 5 or 7 year survival in breast cancer or multiple myeloma or some other cancers, all models work quite poorly. And it's illustrated also on the next slide, which shows performance of Amsterdam 70 gene signature for breast cancer prognosis. It's one of the most important signatures, which is a diagnostic product approved by FDA. So in the original study published in 2002, the specificity of the 70 gene biomarkers was very high, about 90%. But in repeat studies with more samples, the specificity went out quite dramatically, and there is very simple statistical reason for that. So next slide, please. We show that instead of statistical analysis and tools for generation of such gene signatures, we can use the real functional grouping, for instance, the pathways, to come up with classifiers. So you can see how it works. So on the left, we have gene signatures deduced from two independent studies, and you can see that they don't overlap. This is exactly what people observe in gene expression studies. Gene signatures don't cross-talk very well. Nevertheless, these genes could belong to the very same pathway, and although they are not statistically significant at the level of genes, the pathway itself is significant and can be used as a classifier. Next slide, please. This is particularly important in genetic studies like GWAS studies, when very large SNP arrays and population numbers usually do not allow to come up with the statistically significant SNPs. So people who use in this field and GWAS studies learn to use pathways as template for multivariate biomarker 3 or 4 years ago, and there is a large field of study dealing exactly with that. Next slide, please. So eventually, what it boils down to is what we call functional fingerprints, sets of pathways which defined such as a barcode complex phenotypes, such as prostate cancer or lung cancer, diabetes 2, liver toxicity, and so on. So any phenotype can be represented quantitatively as a barcode of pathways of multiple types. Normal pathways involved in phenotype, pathology pathways, and drug action pathways. So we believe what needs to be done for systems biology to become a real engine for analysis of omics data and applications in personalized and translational medicine. So this is my last slide. So now back to you, John. Thank you.
Well, Yuri, thank you for your insights and for those examples of ways to better use and understand omics data for personalized and translational medicine. That was an extremely informative presentation. Thank you very much. Before we proceed, let me remind everyone of our Q&A segment that comes right after the panelists have made their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. Cornelia Polyak, is ready to talk about the challenges and opportunities for the personalized treatment of breast cancer. Cornelia, the floor is yours.
So thank you, John, for the introduction. And today I'd like to tell you about some of our work in understanding heterogeneity in breast cancer and how this poses a challenge and gives opportunities for personalized treatment. The next slide shows that breast tumors are very highly heterogeneous. There is a high degree of heterogeneity among tumors in different patients, which is called the intertumor heterogeneity. Based on expression profiling, breast tumors are classified into major subtypes of luminal, HER2+, and basal-like. And these subtypes not only have different expression profiles, but they also have different risk factors for development, response to treatment, different risk of progression to metastatic disease, and also even when they metastatize, they have different site of metastasis that preferentially occurs. In general, luminal subtype tumors are positive for hormone receptors, respond well to anti-hormone therapy, and they tend to be more favorable prognosis. The HER2-positive tumors, again, are very well treated with anti-HER2 therapies such as Herceptin. The basal-like subset currently is subtype that still has no targeted therapy, and a subset of patients responds to chemotherapy, but 80% of them very rapidly succumb to their disease. And these tumors are the ones that preferentially metastatize to visceral organs and also to the brain. Next slide shows that this intertumor heterogeneity could be explained by two potential models. One of them could explain heterogeneity, subtype-specific heterogeneity, based on the differences in cell of origin of the tumors, such as the basal-like breast cancer, which has very similar profiles to the putative stem cells in the breast. They may initiate from those, whereas the luminal tumors, which are more differentiated and hormone receptor positive, may initiate from a more differentiated luminal progenitor. Alternatively, we can also imagine that the same type of progenitor could be the cell of origin to multiple subtypes, and it's more like the subtype-specific genetic and epigenetic events that eventually determines outcome. And most likely the combination of these two is what's happening, and also probably not all basal and not all luminal tumors have the same cell of origin and progression steps. The next slide shows that in addition to this intertumor heterogeneity, there is a very high degree of intratumor heterogeneity in breast cancer, which means that even one tumor of a patient at any given time is composed of cancer cells with different properties, such as, different molecular profiles and also different response to treatment. This example here from Craig Allred's paper in DCIS, ductal carcinoma in situ, shows that even if we just stain for commonly used markers such as estrogen receptor, HER2, and PVD3, we see that a subset of tumors, about 25%, has high degree of heterogeneity for the expression of these markers, as illustrated here by the positively and negatively stained areas. Next slide shows that what are the possible explanations for this intratumor heterogeneity. It has been proposed that tumors are genetically unstable, therefore there is a very high degree of genetic instability, and this genetic instability combined with clonal selection could explain intratumor heterogeneity. In addition to this, there is also epigenetic heterogeneity in the tumors, such as differences in DNA methylation and chromatin patterns, which includes the more and less differentiated features, such as more stem cell-like and more differentiated epithelial cell features. And lastly, there is also a non-hereditary variability, which is like an adaptation of cell populations and also cell-to-cell heterogeneity, which is basically just due to a fluctuation of expression and signaling pathways. And basically all three of these are present in a tumor, and they are linked to each other, meaning like the same cell population can have genetic heterogeneity, epigenetic, and also non-hereditary variability. Next slide shows one of our studies that we conducted years ago trying to characterize the more stem cell-like and more differentiated breast cancer cells. The more stem cell-like cells were purified with CD44, a cell surface marker, whereas the CD24 was used for the purification of the more differentiated luminal cells. On the heat map on the left shows the clustering of these cell types, the expression profile of these cell types on multiple tumors, and as you see, they very clearly form very distinct CD24 and CD44 clusters. We also saw from the expression profiles that the CD44 cells are more stem cell-like, whereas the CD24 cells are more differentiated, and therefore this suggests that CD44 cells are more expressing invasion, angiogenesis, and proliferation-related genes, so therefore they may be more likely to play a role in the metastatic progression. Next slide. When we use the network and looking at pathways based on our expression data to see what are the pathways that could be differentially activated between these two cell types, we found that many of the known stem cell pathways such as Wnt, TGF-beta, hedgehog, and integrin signaling was preferentially active in a stem cell-like CD44 plus cells. On this figure, those are the ones marked, these little brackets marking those particular components of the pathway that are more active or expressed in a CD44 plus cells. This also suggests that if the tumor is treated with an inhibitor of any of these pathways, then only the CD44 plus cells will respond because those are the ones that have activation of the signaling. To test this, in the next slide, we treated the cells with an inhibitor of the TGF-beta receptor kinase. These were primary cultured cells from pleural effusion of the same patient, CD44 plus cells and CD24 plus cells shown in the left and right-hand side. And what we can see on this figure, that the morphology of the CD44 plus cells changed very rapidly following treatment with the TGF-beta receptor kinase inhibitor. The top figure shows the morphology just in a microscope, and then the beta-catenin, E-catenin, and ZO1 staining shows that the cells acquire this very nice epithelial staining pattern in contrast, the abnormal cytoplasmic staining that they had before. This suggests that the cells basically underwent the mesenchymal to epithelial transition. On the other hand, on the left-hand side, as you see, nothing happened to the CD24 cells because they don't have expression of these pathway components, and also, therefore, they don't react to this drug. The proliferation of the cells and the survival, neither of the cell types was affected by this inhibitor, suggesting that it would not be providing elimination of these cells, but it could dramatically change the phenotype and potentially decrease chance of metastatic spreading. As a follow-up to this study, the next slide shows that we decided to conduct an unbiased shRNA screen to trying to identify other pathways and genes that may be essential for the survival and proliferation of the CD24 or CD44 plus breast cancer cells. Unfortunately, such study could not be conducted in primary tumors or primary cultures derived from tumors because those don't grow very efficiently and also they change phenotype. Therefore, we selected a panel of cell lines that seem to reproduce the phenotype of the 44-24 plus cells. On panel A in the figure shows the gene expression profiles of the CD24-44 plus cells in primary tumors, and we identified about 1,500 genes differentiate these two cell types. And then we took these 1,500 genes and applied them to expression data from breast cancer cell lines, 54 cell lines, and what we found that they split into very nicely into two groups. The genes that are high in the CD24 cells were expressed in the luminal cell lines, which are the blue-colored one. The genes that are high in the CD44 cells were expressed in the basal-like breast cancer cell lines, which are the red ones on this panel. We selected about 7 of each of these cell lines that grow well, and we could infect them well with the shRNA and characterize these further for the expression of CD24-44 cell surface marker, which is shown in panel C. So again, the basal-like cell line seems to be largely CD44 positive, negative for CD24, whereas the opposite is true for the luminal cell lines, which are largely CD24 positive and negative for 44. Panel D just shows the methylation status of a few markers that we knew, again, differentiate the CD24-44 plus cells, and in these cell lines also we seem to reproduce this methylation pattern. So based on the expression of cell surface marker and also the methylation pattern, it looked like that some of the cell lines could be good models to use for this study as they have a profile similar to the 44-24 cells. So therefore then the next slide shows that we conducted this shRNA screen with 1,500 genes, and initially we screened 2 basal and 2 luminal cell lines. In panel A shows the summary of the results of the primary screens. We identified about 80 hits in each of the basal and luminal cell lines. Then these genes were screened in a secondary screen in 14 cell lines, so in a larger panel, and the summary of that is shown in panel B of this figure. The hits that were scored as hits in a basal-like cell lines are highlighted in red, and as you see, we have about 15 genes that seem to be basal-like specific hits. There are some that also score in many basal cell lines, but some luminals record these basal-like hits. Luminal hit, there was really nothing that was only specifically scored in the luminal cell lines. ERB3 was kind of a marginal luminal hit in a way that it scored in all the luminal cell lines and not all of the basals, but in some of the basals. And then lastly, we also had some non-selective hits that were scored in each cell line regardless of their subtype. What was interesting that if you look at the top 15 genes, which are the basal-like specific hits, many of these encode secreted proteins and extracellular matrix components, suggesting that these stem cell-like breast cancer cells make their own niche in a way, and they dependent on this niche. Next slide showed that we validated the shRNA screen result for selected genes by using siRNAs on the panel C on this figure, and also small molecule inhibitors for the hits that we had these inhibitors available. And both kind of experiments essentially reproduce the shRNA screen result. In the siRNA screen, we just looked at whether the siRNA specifically affects the luminal or basal-like breast cancer cells for these selected genes. and looking at cell viability, and they essentially gave the same results that were hits in a basal-like cell lines based on shRNA were also hits by siRNA. On the small molecular inhibitors, we determined the GI50 for each of the 14 cell lines, and they plotted them according to decreasing GI50. And again, the red colors indicate the basal-like cell lines, and as you see, many of the hits are actually preferentially inhibiting the basal-like cell lines. The JAK2 inhibitor and the CXCR2 inhibitor were particularly interesting, and we followed up more the JAK inhibitor in detail. The reason is because there are already compounds inhibiting these pathways that are in clinical trial for other indications and therefore could rapidly be tested in breast cancer, and our eventual goal was to actually conduct a clinical trial based on these results. So we focused on an IL-6 JAK-STAT pathway in a further detail. The next slide shows that we actually found that in a basal-like breast cancer cells, there is an autocrine IL-6 JAK-STAT activation loop, first because we showed that IL-6 is constitutively secreted by the basal-like breast cancer cells based on ELISA that shows in this left panel, and this leads to constitutive phospho-STAT3 activation as shown by the Western blot on the right-hand side, and this can be inhibited by a JAK kinase inhibitor as shown in this lower panel. The next slide shows that we decided to test if we can inhibit the growth of the basal-like breast cancer cells, which are CD44 plus and phospho-STAT3 plus in vivo in xenografts. So therefore, we derived xenografts from the 4 basal-like breast cancer cell lines indicated on this graph, and also a patient-derived tumor, IDC31, which had a phenotype of largely composed of CD44 plus, CD24 negative, and phospho-STAT3 plus cells, as shown in this little upper inset here. What you see on the figure is that JAK kinase inhibitor effectively inhibited the tumor growth in all of the cell lines and also in a primary tumor to some degree. The significance of this was variable, and that was partly due to the fact that some of the tumors were very small because the cells are not very tumorigenic. But even in these tumors, like the HS578 and IDC31 xenografts, when we looked at the cellularity of the tumor on the histology on this H&E same slide, we saw a very dramatic decrease in cellularity following treatment with the JAK kinase inhibitor, which is this BSK805. And on this right graph just shows the quantification of this decrease in cellularity. Next slide shows that we looked at the frequency of the phospho-STAT3 positive tumor cells in the xenografts derived from the cell line. The SUM185 cell line is 100%, 44 plus phospho-STAT3 plus as shown on the control treatment slide. And following treatment with the BSK805 inhibitor, the number of phospho-STAT3 positive cells dramatically decreased. The same was true on the IDC31 as shown in the figure, and then this is actually quantitated in this graph on the right-hand side. And we also noticed when we looked at the histology of the tumors that they actually seem to have, the tumors after treatment that responded, seem to have decreased number of leukocytes, infiltrating leukocytes, and also less angiogenic, suggesting that this JAK inhibitor may decrease tumor growth multiple different ways. The next slide shows that we actually wanted to further test the importance of STAT3 in these basal-like breast cancer cells, so therefore we derived shRNAs against STAT3 and expressed these in a SUM159 basal-like breast cancer cell line. And when we derived xenografts from these cell lines, we found that there was a delay in tumorigenesis, so there was a higher fraction of mice that survived without tumors in the shRNA-expressing clones. And then even the ones that eventually developed tumors, they seem to have escaped the shRNA effect. As shown on the right-hand side, they basically re-express STAT3, which suggests that there is a very strong selection for STAT3 expression. The next slide shows that when we looked at the network formed by the 15 basal-like specific hits, it was interesting that each of the hits that are highlighted in red rectangles seem to fall into one network, and it looks like that STAT3 may be a common downstream target of multiple of the pathways that are important for these cells. We tested this experimentally. The next slide shows that basically we found that the phospho-STAT3 levels seem to go down following treatment with inhibitors to other pathways on the left panel, and on the right panel also we show that the STAT3 transcriptional activity seemed to go down in a HS578 basal-like cell line following treatment of these inhibitors, which are not directly targeting STAT3 pathway, but looks like that eventually they seem to affect transcriptional activity. The next slide shows that we also derived a STAT3 gene expression signature that seemed to predict outcome in breast cancer patients. The left-hand side shows this signature derived in a HS578 basal-like breast cancer cell line. Basically, it looks like that the patients who have the tumor expressing this signature seem to have shorter distal metastasis-free survival. This is the red graph here. Whereas if we derive the same signature in a MCF7 cell line, which is not a constitutively active STAT3 cell line, this signature was not prognostic in a patient. Next slide. And lastly, we also examined the frequency of phospho-STAT3 positive cells in human tumors, 170 primary breast tumors of all subtypes we looked at, and we also looked at the expression of CD24/44. On the left panel shows representative examples of staining, and we found that the majority of the CD44 plus CD24 minus cells are phospho-STAT3 positive, whereas CD24 positive, CD44 negative cells, which are these blue ones here, they almost never phospho-STAT3 positive or very rarely. The right panel actually shows the quantification of this, and as you see again, like the 44 plus 24 minus cells are very frequently positive for phospho-STAT3. The next slide shows that this happens actually in every tumor subtype. So basal-like HER2 plus luminal and A on B tumors, the 44 plus 24 minus cells seem to be positive for phospho-STAT3 irrespective of the cell type. But then the frequency of the 44 plus cells is the highest in the basal-like tumors and lowest in the luminal tumors. So at the end, it still looks like the basal-like tumors are most likely to have these cell types. The next slide, just a summary of what we think is how these autocrine and paracrine pathways could be functioning in the tumors. We think that the basal-like CD44 plus 24 minus stem cell-like cells have this constitutive activation of the phospho-STAT3 pathway due to the secretion of IL-6 and also because of the activity of some of these other pathways that eventually seem to be activating STAT3. In the CD24 cells, this pathway is mostly inactive, but sometimes it can be turned on due to paracrine signaling from the neighboring stem cell-like 44 plus cells, or inflammatory cells or stroma could express some of these cytokines. The last slide shows the conclusions that can be drawn, that basically we see that the cancer cells display very high genetic, epigenetic, and phenotypic diversity. All of these can change during treatment, but it looks like that despite this high degree of heterogeneity, maybe we can find specific pathways on which these cells dependent on, such as the JAK-STAT signaling pathway for the stem cell-like breast cancer cells, and that could be explored as a future therapeutic target for a subset of breast cancer patients. And the last slide just shows the acknowledgement of the people who did the work. Vanessa Almendro, Lauren Marotta, and Mikhail Shipitskin did the major part of this work that I just presented, and we had numerous collaborators in other institutions and also within Dana-Farber. And back to you, John.
And thank you, Cornelia, for your very detailed description of the new ways to approach the treatment of breast cancer. And in following your slides, I really think that many in our audience are going to find the information you presented very useful in their own research. Very good, thank you. I want to welcome Dr. Craig Webb back to the webinar speaker lineup. Actually, Craig's never really left the webinar. He's been listening to the other presentations, as have we all. But he's now set to give the second part of his talk. Craig, you can begin part 2.
Thank you, John. All right, so I'm going to pick up where I left off, having explained the infrastructure we built, tell you about some of our early examples of real-time personalized medicine trials, as well as touch on the future, where I think this is going. All right, so how do we help today's patients? This, by the way, is a 3-year-old first patient on our trial, 3-year-old with metastatic neuroblastoma, so no question to motivate yourself here on this one. So this patient's obviously failed multiple lines of therapy and really is coming to the end of the road where you try something or hospice type of situation. So next slide. These are the people involved, many people to thank, not just myself here in my lab. This was many disciplines across institutional and political lines and many great people that we had a chance to collaborate with. Next slide. So the general challenge was this has to be real time. This cannot be stick it in the freezer and come back to it in 6 months. How do you enroll patients on trials, collect a tumor specimen, qualify it through pathology, move it into a clear lab, the CMM that I told you about in the first part there, run it through, in this case, the Affymetrix technology back then, run it through the algorithms that we developed, which I explained. Create one of these actionable reports and provide that back to the physician, and our challenge was do that in 5 business days. So it's really more of an engineering feat, a logistical workflow issue than it was a scientific issue at that time. Now, on the bottom half here, you can see we developed these tumor models. They're called primary tumor graphs that we use for vetting new ideas and new combinations of agents, etc., which I'll describe briefly as well, but very, very briefly. Next slide. This is a picture of some of those tumor graph models. We have over 120 of these models now that very closely resemble the patient's disease at the pathological level there on the left, classical IC, but also at the molecular level. We gene expression profile all these. We also look at target mutational analysis of certain key actionable mutations that we have. And these models very closely resemble, in fact, perfectly resemble the patient's disease over about 3 or 4 in vivo generations. So we use these models a lot for our research, and we have these available for others across 35 different pathologies if anyone's interested. Next slide. Okay, getting back to this 35,000-foot view, I'm sure many of you are thinking, what's in this black box, which I'll now describe. Next slide. So this actually is part of a FDA-approved IDE application. So believe it or not, we've gone through the FDA with this quote device. And basically, a tumor specimen is compared to a reference set. It depends on the tumor type that you're interested in. And then a differential gene list, we use Z-scores, which are standard deviations from the mean or the reference set, go through a series of algorithms, including, if you notice there on the sort of vertical, is a GeneGo system. So these are some of the network-based analysis that Yuri described. The hidden nodes methods, for example, are in there. But we also use some signature-based methods like the connectivity map and gene set enrichment analysis, and then simplistic rules-based methods that we developed, simple logic rules that if a drug target is overexpressed or mutated, then give drug, for example. And then each of those methods can predict either targets and/or drugs. They get mapped together, and at the bottom here, you notice where it says association with public clinical contextual knowledge. That's a key part, because all we're doing here really is predicting molecular efficacy. It's a molecular hypothesis of drug efficacy. We then align with real-world evidence that may support that hypothesis. In our clinical trials, we then have to go through a tumor board that discusses these reports and decides the treatment plan for the patient. Next slide. Here's an example of the report. It's a list. In this case, this is a summary view of the report where it lists drugs top to bottom for predicted efficacy. It lists the methods that are used to predict the drug efficacy. And then it aligns, as you can see on the right hand here, supporting evidence. Now, in the world of community oncology, that people care about reimbursement of the drug. And as you know, reimbursement is really indication-specific at this point. So even though there are multiple lines of evidence suggesting veronostat may have worked for this patient in this disease context, there's no evidence supporting its use that can be used to reimburse a drug. So physicians may typically gloss over that one. But down here, for number 4, for example, we can see that there is evidence for that. So now on the right-hand column, for example, there's clinical evidence that includes clinical trials that are currently looking at the drug in the context of the patient's disease. Next slide. And for that evidence, we actually are using multiple knowledge sources, but it includes Thomson Reuters, for example, DrugDex or Micromedics, for example. So this is just looking at evidence for Avastin in breast cancer, which is a topic of debate right now, but here's the current evidence that supports the use of Avastin in breast cancer. So again, we align these molecular predictions with evidence that is relevant and practical for the treating physician. Next slide. So going back to our first 50 patients, we did a 50-patient trial just to work out whether we could do this in 5 business days. The answer, of course, was yes, we could. But you can tell from the pathologists here, we went across pathologies, hematological and solid, went from age range 3 to 80, so both pediatric and adult, and basically worked out the optimal processes within the lab, had some process failures, which we've corrected most of those. And then we tracked whether physicians used the information, and then also anecdotally whether or not we saw any responses. So I'll just summarize that. Next slide. Here's one example, actually, with Dr. Lester out of St. Joe, Michigan, a patient with non-small cell lung carcinoma, again, multiple relapse refractory patient's disease. Picture of the biopsy on top left here. And this is just a visualization back then of how we visualize these networks. And I've highlighted EGF receptor in the middle. You can see the red, what we now have thermometer bars, a red dot on the EGFR that was overexpressed and suggested to be amplified at the chromosomal level based upon some methods we use. And then that had to be confirmed by FISH. That allowed the physician to get reimbursement for olatinib. This was back then. And this is what the reports used to look like back then. You can see some of the EGFR inhibitors listed here at the top of the report. And this is how the patient responded to a combination of all molecular guided therapies, cisplatin based upon low ERCC1 levels, docetaxel, olatinib, and Avastin were all given to this patient, and the patient showed a good partial response to that combinational approach by PET, CT, and also biomarker levels. Next slide. So our conclusions from that's just one example. Conclusions from the first 50-patient study was, the objective was, is it feasible in a 5-day business day turnaround? And the answer was yes, it was. Were the reports deemed to be useful? Yes, we actually tracked how many, 24 out of 34, I think it was, used the information, and we addressed why the others didn't. And frankly, it came down to the readability of the reports, and so we've obviously improved on those over time. And then we saw some responses that certainly provided us encouragement to keep pushing this area forward, and these were presented at AACR by Drs. Lester and O'Rourke a few years ago. Next slide. So just to let you know what we're doing currently. So we have a number of these feasibility and safety trials ongoing, one in pediatric neuroblastoma specifically. There was a big announcement recently that Dell actually have got behind this and actually developing some cloud infrastructure around this, which I'll touch on briefly. Believe it or not, we're doing the same for canine trials, veterinary oncology trials, and we're working with the COTC from the NCI, but also osteosarcoma trials, for example, with Pfizer. We are working with third-party payers who are very interested in this. Does it reduce healthcare costs associated with end-of-life off-label use, for example? That is, as you know, not particularly molecularly informed at this point. And we are working with the company Intervention Insights as well as Priority Health in Grand Rapids to work on those trials. And then we have efficacy trials, and these are the ones that we focus on scientifically in metastatic melanoma, glioblastoma, non-small cell lung carcinoma, and again, pediatric neuroblastomas that are either ongoing or in development. And this is where we will assess head-to-head with current practices, standard of care, whether or not molecularly informed therapies actually improves the outcome of these patients in large randomized trials, and that's where it has to go. So quickly, next slide. This is just a synopsis of the pediatric neuroblastoma trial. This is the one where we actually sought IDE approval, and we obtained IDE approval from the FDA. Got an IND waiver because we're only using drugs that have been previously looked at and have pediatric dosing here. And that's the clinical trial number if anyone's interested in looking that up. Next slide. It's actually, as I mentioned, is being sponsored as well now by Dell, and they're developing the cloud-based infrastructure to allow this really to scale beyond 14 patients was that first trial, and allow the physicians, there's 16 sites now involved in this trial through work of Giselle Scholler at the NMTRC, who's developed this consortium of pediatric hospitals who are collectively working on these sorts of personalized medicine approaches. Next slide. So finally, this is the future. Next slide. Again, patients are still there. Patients will still present with metastatic disease for some time to come, probably more. The drug pipeline on the right is getting bigger, so there's certainly a need to be better with the drugs that are already there in existence. The knowledge is expanding. I think our knowledge is just represented by a GeneGo plot of actually of the existing pharmacopoeia, all existing drugs and the mechanisms of action that shows you how much redundancy there are across company pipelines. And then, of course, the technologies that are continuously evolving and are moving us into this area of next-generation sequencing, which is the area that we're focusing our efforts in now. So, next slide. There's efforts to bring next-generation sequencing down to a cost and time-effective point. David Craig at TGen, for example, is heading up some of these efforts with the 1,000 Genome Project. So, next slide. And there's immediately, this is RNA-seq data, and that's our sort of first effort, but there's immediately information that you can glean from a classical RNA-seq project, not just at the expression level, of course, which replaces the need to do a microarray, but also gives you some expressed mutational data. In this case, we're identifying a RAS mutation in a patient's tumor. And of course, next slide. If you have a RAS mutation and you happen to have colorectal cancer, there's already guidelines that suggest you should not take cetuximab in this case. Next slide. So the last slide here, then Dr. Daniel von Hoff, who works at TGen and works with us on many projects, but who is really pushing the envelope of using personalized medicine in the context of phase 1 trials. If you align the mechanism of action of the drug, in this case a smoothing inhibitor, with the molecular vulnerability or context of vulnerability of the patient's disease, in this case a mutation in patched1 or a hedgehog pathway, you can start to see spectacular responses even in the phase 1 setting, which of course has not been what they've been designed for, but more and more you'll see that being used. Next slide. So last slide, many people to thank, too many individuals to thank, and therefore I typically thank institutions or organizations at this point, but including Jingo Thompson-Reuters, who have really been wonderful collaborators over the years here for us. So that concludes my slides. Thanks, John.
And thank you, Craig. And I want to extend my appreciation to you and to the entire panel for your excellent presentations. In fact, I feel like I've just taken an up-to-date course on genomics and personalized medicine, so thank you all. And it also looks like you've all struck a chord in our audience as we received a number of great questions from them. So let's get started on our Q&A session. And the first question is for Craig. What methods exist or could be developed to predict optimal combinatorial therapies?
Okay, and you can hear me okay, I assume?
Yeah, Craig, you can answer.
Okay. Yeah, that's a great question. So, in fact, we today actually announced a Stand Up to Cancer grant in metastatic melanoma, addressing this in part. We do believe there's some systems biology tools that Yuri alluded to, the hidden nodes methods, for example, where if you imagine now looking at the system-wide, you're trying to have a maximum disruption on a tumor-specific system. The key is not to be redundant within linear pathways, but to go across non-linear pathways, if you like. So if you look at that point of divergence within the system space, we think that's one approach that we're going to try and develop for a smart combinational therapy selection. But we're also looking at some other methodologies, such as the connectivity map out of the Broad Institute. If you imagine drug A has a genomic effect and drug B has a genomic effect, and now can you simply add those genomic effects to get a combinational therapy? So we're looking at multiple methods for that, but they're not developed yet. I think that certainly is the future of oncology treatment. We all probably know that combinations is the right answer. So, great question.
I got another great question for you, Craig. What is the biggest problem in the translation of whole genome sequencing into clinical practice?
A whole genome sequencing. At this point, whole genome sequencing, which is the full genome, probably time and money. At this point in time, it still takes too long for real-time decision support, at least in the oncology care world, to move through a whole genome sequencing approach. Now, having said that, that will come down. I think some efforts to, for example, bring instrumentation into a cloud-based environment to stop the time it takes pushing data around the pipes between institutions is probably a major rate limiting factor. So that will get better. But there are, as you know, more non-whole genome approaches like custom content, next-gen sequencing approaches, things like Ion Torrent, for example, that are moving their panels into clear labs as we speak. So I think that will be the first inroad for whole genome, not whole genome, but sort of targeted sequencing and maybe exome sequencing.
I'm really impressed with these questions today, Craig. I have another one for you. Is it realistic that omics technology will be installed in departments of clinical biochemistry, or must biomarkers be translated into conventional assays before clinical evaluation?
Well, that sort of kind of hits, it's another great question, hits the regulatory discussion that's ongoing, as you probably know, between the FDA and organizations like CAP, for example, talking about whether or not these laboratory developed tests, lab-based tests like LDTs versus FDA-approved tests. I think the jury's still out where that's going to end up. We've kind of kept our options open, I guess, and we've actually, as I mentioned, filed an IDE for a gene expression device, but we also have run laboratory-based tests as well. So I think the verdict's still out there. Now, the question is, can labs do that? Can hospital labs, for example, develop their own tests? Absolutely. Absolutely can. I think some of the technologies, as I mentioned, things like Iron Torrent or MySeq are coming through, which will make that much more doable than full genome, high-seq type of approaches. So.
And Craig, will NGS be able to substitute all DNA and RNA assays in medicine in the future? If yes, when will it happen? And crystal ball time. So if you can answer that question.
I hope so. So I would say yes. When? I think optimistically, I think certainly within 10 years, and I would hope within 5 years.
And Craig, we have another question. Does sampling of tissue affect the heterogeneity, normal versus tumor margins?
Absolutely, yeah, absolutely. Of course, in fact, the FDA approval process for our IDE requires us to take multiple samples looking at the heterogeneity of tumor biopsies, for example. And the good news is that as long as you hit the tumor and you qualify that using pretty stringent criteria, looking at the percentage of tumor versus percentage of necrotic or normal tissue, we can get pretty reproducible results. So, but yes, we have to be very careful with avoiding, obviously, missampling.
Thank you, Craig. And Yuri, I have some questions for you. How does GeneGo interact with next-gen sequencing data? Is this a cloud-based application? How is it priced, and is there a subscription involved?
Well, we do not deal with next-generation sequencing data per se. It's a bit different expertise and specialty. We actually collaborate with Genome Quest, one of the company's leaders in next-gen sequencing analysis. What we have as an input is just a list of genes, or the list of SNPs, for instance. So we collaborate with Genome Quest and also with Illumina.
And Yuri, if you have a potential drug compound that is very effective for certain cell lines, but not effective for all others, or effective at all for others, what methods would be the best way to identify drug targets or biomarkers?
Well, we actually have several projects like that with pharmaceutical companies. We do it as service. So essentially, we have the data from Researchers Onco panel, where more than 200 cancer cell lines are either resistant or sensitive to certain cancer drugs. And we usually use expression data or genetic data coming with these cell lines. So when we have expression data, first we calculate the cell lines which are sensitive and resistant specifically, so it's correlation with EC50 and EC50 data. And then we apply functional analysis, actually using MetaCore and a couple of custom scripts to come up with what we call causal networks. So essentially we build networks for resistance and network for sensitivity, and this is the source of potential targets and biomarkers. So you can analyze causal networks in many different ways.
And Yuri, we have another question. How can I design or discover a pathway from gene expression microarray experiments?
Well, first of all, a great number of pathways are already discovered, and they are kept in pathway databases such as KEC, such as MetaCore. Like, for instance, we have more than 1,200 pathway maps. Each has 3 or 5 pathways, metabolic transport pathways, signaling pathways. So first what you do, you just do enrichment, and you look for the pathways which are already there and involved in your expression data set. So you can start with a subset of genes which would hit certain pathways from cell cycle apoptosis, and then you can build networks. Or you can start building networks and do all kinds of interactome analysis on your own, and then try to design the pathways using tools. We have a couple of editing tools. But the pathways, in our definition at least, is something that is experimentally confirmed. So what you can generate using the networks, interactome, for instance, is just a hypothesis. So whatever you designed has to be experimentally validated as a multi-step pathway.
Okay. And Yuri, another question. How can I use GeneGo for the pathway analysis for time course microarray expression data?
Well, it's a standard use of metacore, multiple time points or different patients, different cell lines, and so on. Well, you simply upload several of them. It can be probably up to 12. It's relatively straightforward to use, and you can apply the workflows. And standard workflow would identify common genes and specific genes, unique genes, similar genes, and then run analysis using networks and enrichment and ontology tools. So that would be the first start. Or you can do it manually, and we have a couple of tools which can be very specific on differences and common parts between the experimental data sets.
And does Metacore provide functional fingerprints of in vitro models of cancer, such as cancer cell lines?
Not really. So for functional fingerprints, you really need to have some statistical analysis done on multiple large data sets, preferably more than 100 samples. So we have tools to derive, functional fingerprints. For instance, find the distribution of pathway maps or build a network, but then they have to be validated, and it cannot be done yet in Metacore. So it has to be a bit different study. So we do it actually using R and bioconductor and some statistical tools.
Thank you, Yuri. And Cornelia, we have some questions for you. Epigenetics is important in shaping the clinical course of the disease. I'm not sure basing therapies based on omics is going to pay off. Do you have a take on this?
Yes, so I already answered online, but omics includes epigenetics, so I'm not sure what the question is, because there are many omics studies in using DNA methylation patterns and also histone modification patterns. So actually the integration of all that into networks and pathways is what's really can be powerful. So you look at gene regulation as many different levels, expression, epigenetics, and so on. So I think that will be actually very useful, and it's already used for this kind of classification of tumors.
We have another one here. Who will be looking at the biomarkers, the pathologist or the oncologist? I know they work as a team, but a lot of research is going on now in tumor markers for diagnosis.
Okay, so here oncologists are the ones that are ordering the tests. So I would predict that the oncologist would be one making decisions, but of course they will collaborate with the pathologist. In terms of who would be looking at a biomarker, actually doing the assay that can be done in a pathology lab and assessed by a pathologist, or also there is a trend, some of these more complicated assays are done in a centralized lab, like Oncotype DX type of assays. You just ship the slides to a company who does the test and gives you back the result, and that ensures that everybody who has that test get it done the same way, and there's no variability in pathology labs. So, but at the end is the oncologist who's managing the patient care, so they would be the ones who decide what assay to order in terms of which biomarkers to look at also.
Okay, and Cornelia, how can network pathway analyses be used to predict cancer cell dependencies on their environment? For example, paracrine interactions are important for survival.
Okay, so this kind of approach hasn't been really explored, and it's actually pretty complicated because you would need expression or signaling data in individual cells after they interacted. So you would have to know which cell has what pathway activated, and then based on that pattern, then you could predict what are the interactions. So, for example, if you have a breast tumor, and then you would be able to sort out stromal and epithelial cells, and then you would see stromal cells secrete a ligand, and then the pathway is activated in a cancer cell. That would be indicative of an interaction. But otherwise, if you're doing the profiling on bulk tissue samples, then it's really difficult because you don't know where the signal is coming from. So for this kind of studies, sorting the cells would be really important.
And Cornelia, let's see, this Another question. We have a number of them here. I'm trying to pick the right one. Here we go. Do you have to account for different isoforms of CD44 from alternative splicing?
Well, it depends on the question. So what the type of screen we did when we were just looking at cells that seemed to have more mesenchymal phenotype, the particular isoform was not important, but if you for example, want to target a particular isoform, then it would be important to know, and that is a very complicated splicing of CD44 that can be very cell type specific, but this was not really relevant to our study.
Well, so I want to thank the panel for answering all the questions, but unfortunately we've run out of time. But please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. Thanks again to the panel for their outstanding presentations, and I want to say thank you to our audience for your attention and for your very thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Thomson Reuters, who made this event possible. Shortly, we'll be sending you a survey, and we would appreciate your feedback on this webinar. Please look out for it and kindly give us your thoughts, as this will help us to continue to bring you topical and timely webinars in the future. Thank you very much. Have a great day.